This DVD is composed of films that were to commemorate the 50th anniversary of the Microcirculatory Society and was presented at the celebration of the 50th anniversary of the Society in Washington, D.C., April 9, 2004. The first film is by August Krogh, who won the Nobel Prize in 1920 for his work on microcirculation in capillaries and small blood vessels. It's an amazingly elegant motion picture considering when it was made in the late 1919 and 1920. Most of the images are from capillaries and blood vessels in the frog, and for that reason the red blood cells are elliptical in shape and are nucleated, as you can see in this scene. The next sequence is also of the frog lung. This is a picture more or less what Malpighi would have seen in the 1600s when he looked at it with a magnifying glass. It's also an example of sheet flow as has been described previously. This a little higher magnification illustrates the deformation of the erythrocytes as they pass through this extensive capillary network in the lung of the frog. It should be noted, however, that frog erythrocytes, because they're enucleated, are not as plastic and flexible as are those of mammals which have nuclei. This shows some stimulation of the blood vessel, small vessel in the nictitating membrane, and you can see all the changes in flow as the diameter of the vessel changes during stimulation. These, of course, are mammalian red blood cells, which lack a nucleus and are biconcave discs. This film was filmed by the Clarks at the University of Pennsylvania in the 1930s and illustrates the responses of the blood vessels regenerated in the ear of the rabbit in the rabbit ear chamber to cold. The decrease in temperature was provided by going outside and taking some snow and packing it around the ear of the rabbit. And as the temperature changed, as the snow melted, they looked at the reestablishment of blood circulation as indicated by the subtitles. Most of these early films had graphic as well as textual material included in them, and as you will see, telling you what is coming in the subsequent scenes. And that's the case with this one. Now as the temperature approaches 97 degrees, or body temperature, the flow is quite rapid and back to essentially normal conditions. With the removal of heat, an inflammatory reaction begins to take place, and white blood cells begin to stick to the endothelium of these vessels. This may well have been some of the first recordings of a microvascular inflammatory response. And with time, increasingly larger numbers of white blood cells were sticking to the endothelium. The following scene shows examples of vasomotion. What also can be seen here is the evidence of streamlined flow in the larger arterial in particular, with the light area, and the peripheral plasma layer you can see at the edges. But it's a very good example of vasomotion in these vessels. The next film is produced by George Fulton and Brenton Lutz at Boston University in 1941, and shows examples of studies of the neuromotor mechanisms in small blood vessels in the retrolingual membrane of the frog. These may well be some of the earliest examples of neurostimulation and examination of the function of nerves on the control of blood flow in the microvascular system. There are examples now will be shown of some of the parasites or Roget cells, as well as muscle cells that surround these vessels, which were thought to be the contractile elements. These cells have been stained with methylene blue in vivo. Here the plexus of nerves can be seen. It's a little bit faint, but I think you can see this as the camera scans the microscopic field. It's quite a plexus of nerves around these blood vessels. The following will be examples of stimulation of these nerves and the subsequent dilatation and constriction exhibited by the vessels. The electrode can be seen over to the right side of the field. So another example, the electrode is to the left of the arteriole. There's the dilatation. And subsequent contraction of this small arteriole. The next film was produced by Melvin Knisley and his associates in 1944 and represented a several-year study at the University of Tennessee during the Second World War on the microcirculation and its changes to infection with malaria. It's one of the first movies that was made in color of microcirculatory phenomena. It should be pointed out that the complete film is about 1,200 feet in length, and what is shown here is only small samples from that film. should be pointed out that Melvin Knisley was a student of August Krogh, and that's why this tribute is in this film. Knisley developed the fused quartz rod technique for looking at living organs in situ in their normal anatomical position. This first scene illustrates the quartz rod, which is extending from the right-hand side of the screen and is inserted underneath the great omentum. The tissue then was observed usually with a stereobinocular microscope, although occasionally a compound microscope was used. This shows a very young Edward Block looking at the mesentery of the monkey. This is relatively normal microcirculation with an arteriole on the right side and a venial on the left, flowing through the adipose tissue in the mesentery. This film and the existing studies represented here were used to probably make some of the first determinations of what constituted normal, healthy blood flow. In other words, no individual red blood cells are seen flowing, no sticking of white blood cells to the endothelium. Flow is in a continuous fashion. This shows normal flow. You can see the clear axial stream, which is indication of streamlined flow, both in the venial, and it's a little harder to see it in the adjacent arteriole. If one speeds up the filming rates to get to some slow motion, you can see the streamline effect here with the red blood cells on the periphery flowing slower than those in the axial stream. And also the existence of a cell-free peripheral plasma layer adjacent to the endothelial wall. An example of normal flow. And on the left side of the venial, adjacent to the fat cells, there is a lymphatic, which is difficult to see, but you can see the two parallel endothelial walls. And external to the left of that, then there is a nerve which is taking a somewhat circuitous course along next to the capillary. Cast to normal, during malaria, the red blood cells begin to clump together to form aggregates, leading to the formation of sludged blood flow, a term coined by Knisley after watching these reactions. Again, here you can see the aggregates of red blood cells. White blood cells are sticking to the endothelium, indicating an inflammatory process. In contrast, the following scene is of normal flow. It should be pointed out that during sludging, these large aggregates frequently begin to settle to the bottom of the vessels, which is the basis for the term sludge. This is the extreme example, as the animal is nearly dead at this point, showing large clumps of red blood cells, some of which are settling. Notice how the fat cells have separated due to the edema that is formed to the extravasation of fluid through the leaky vessel walls. Notice as these clumps of cells flow from left to right, they become closer and closer together due to the loss of fluid through the vessel walls. The next film is by Chambers and Zweifach, produced in 1946 at New York University. Again, the frog was the animal of choice for these studies. The captions are relatively self-evident, so there are only a few points here where I'll comment on these pictures. Shows an arteriole and flow into a capillary. The flow being collected into a venial. And into a larger venial. Note the rolling of some leukocytes along the wall in this venial that's illustrated in this picture. It's a very nice picture showing a rolling of a single white blood cell along the wall endothelium of this venial. It should be noted that the white blood cells in the frog are considerably smaller than the erythrocytes, in contrast to mammals. An example of some white cells tethering to the endothelium prior to diapedesis. The next film was produced by Edward H. Block in 1948, the University of Chicago. And these were some of the first studies of the human microcirculation using the bulbar conjunctiva as the site for observation. The first scenes show the technique in using a small lamp to direct a beam of light to the bulbar conjunctiva, and then using a stereobinocular microscope for observation of the patient's conjunctival circulation. This is an example of the equipment used that was taken to the bedside, the microscope mounted on a special stand, and a 16 millimeter camera that was compact and could be mounted on the microscope for recording of the human bulbar conjunctival microvascular circulation. Using this technique, Block studied a number of disease processes and recorded them on motion picture film. This film was made by Nickel and Webb in 1948 at Indiana University, and is some of the first recordings made of the bat wing, which Nickel and Webb and some subsequent investigators have used as a good site for studying mammalian microcirculation. An example of a capillary feeding off into from an arterial. And you can see a little bit of vasomotion there, spontaneous dilatation of this area of the, might be called the precapillary sphincter. It's an example of vasomotion in an arterial at the top. The reason for the pulsatile flow in the venial at the bottom is not clear. But there's considerable changes in diameter in the arterial spontaneously. Very nice pictures showing vasomotion, changes in diameter in these small blood vessels. The next film was produced by Edward Block in 1960. At this time, he had moved to Case Western Reserve University. And these are the earliest high-speed motion picture analysis of microcirculation as far as I know. The analysis was done both in mammals as well as in frogs. The technique used a Kodak high-speed motion picture camera with a rotating prism for a shutter rather than a mechanical shutter, as illustrated in this diagram. It attained speeds of up to 3,000 frames per second, and as a result, it took 100-foot spools of film, and during the first 90 feet, the camera was just getting up to speed, so the only part of the film that was used for analysis was the last 10 feet, which was developed by hand immediately after the experiments. This shows the camera imaging through a microscope set for a vertical mesenteric preparation using the quartz rod. This is a vertical mesenteric preparation of a frog on a microscope is mounted on a horizontal optical bench. Here's the preparation. And the following are some of the images obtained. This is normal speed. And these are negatives taken with Tri-X high-speed film, showing both a arterial and a venial in the frog mesentery. And you can notice particularly in the arterial, you can see the changes in the distribution of shapes and so on of the red blood cells. All of this was analyzed on a frame-by-frame basis. There's another example of higher magnification showing the bifurcation of two vessels and the red cells sliding off, sandbagging around the bifurcation and sliding off one direction or another. Notice how some of the red cells are stacked up like in Rouleau formation, while others are at right angles or more acute angles. This was all analyzed on a frame-by-frame basis on a stop motion movie projector with projection on the wall and a lot of drawings to analyze some of these flow patterns. The flow appears to become slower and slower as the camera is getting up to maximum speed. And this is the normal imaging of the microcirculation. This film was made by Dr. Block and myself in 1962. And it was the first attempt to try to use the television system as a scanning microdensitometer. By scanning one line, one raster line, which could be selected anywhere on the TV screen and displaying the voltages across that line, you could use this as a scanning microdensitometer. This is an example where the tissues are transilluminated with monochromatic light at 414 millimicron, and you can see the absorption of the hemoglobin. In contrast, if you go out in the green, there's a minimal absorption, and you don't see an absorption peak on the oscilloscope. There are two black bars, vertical black bars, and this was used to set the black level on the oscilloscope trace. Again, back at 414 millimicron, you can see the absorption peak in the center of the oscilloscope trace. This then was used to try to image and get some time measurements of the distribution of a dye injected into the bloodstream as it passed from the bloodstream out into the tissue. To obtain this, the movie camera had a split lens so that we could film the oscilloscope and the movie screen at the same, or the TV screen at the same time. Now a dye will be injected, which is absorbed. You can see the peak as the dye goes through the blood vessel. And as the dye passes through, the peak diminishes. What's not particularly clear on this particular example is that subsequent peaks, there's another injection of dye, and then one begins to see peaks along the representing areas outside the endothelium as the dye moves into the tissue. You can see a little bit of this on the right side of the main peak on the oscilloscope trace. This last film was made by Dr. Block in 1963 to illustrate some of the things that could be seen by in vivo microscopy in various organs, specifically the lung, liver, and kidney. Again, this was made at Western Reserve University in Cleveland. first scenes were of some glomerular circulation in the frog kidney. Where you can see the glomerular tuft, Bowman's space. And this is a televised image of the same thing. These are glomeruli that are in grafts of kidneys in the hamster cheek pouch chamber. Following are examples of the microcirculation in the liver of the frog. Both with a video microscopy as well as direct color imaging with transillumination with white light. Using a wavelength in the green, one could get fairly good cellular detail. This is illustrated by several hepatic parenchymal cells and their nuclei, as well as some of the organelles within those cells. This illustrates how some of the endothelial cells can act as sphincters in the sinusoids of the liver. And finally, a scene of the lung, as shown by Malpighi in 1661. We saw an earlier example of that from the Krog film. This is an example of the frog lung taken by white light transillumination and color filming. This is an example of mammalian lung, rats, taken with the video system. And again, frog. And you can see here some of the epithelial cells, some of the organelles within those cells, as well as the blood flow in the very dense capillary plexus in the alveolar walls of the frog lung. It should be noted that many of these films are available for viewing at the National Library of Medicine in Washington, D.C., and we thank the National Library of Medicine for allowing us to make copies of this for this presentation.